Welcome everyone, welcome Janilia and welcome EMBL. With today's talk, we are kicking off a new seminar series, which combines for the first time Janilia's and EMBL's optical interest groups once per month. Within the next upcoming 6 months, you will be able to broaden your horizon with exciting talks about optical imaging, dye development and image analysis, alternating between scientists at the Janilia Research Campus in the US and the EMBL in Europe. The aim of the combined seminar series is to foster the exchange of ideas and network between our two institutions. At Janilia, the seminar series will be hosted by Chen Wang and me, Ulrike Böhm. And at the EMBL, the seminar series will be hosted by Robert Prevedel, Dimitri Krom, and Christian Tischer. Today's speaker is Kasper Podorowski, where his lab develops hardware, algorithms, and indicators for imaging neural activity in vivo. He grew up in Toronto and did his PhD studies in Vancouver, Canada. In today's presentation, he will talk about how programmable microscopes can improve activity imaging. So welcome, Kasper, and please feel free to share your slides and take it away.
Thanks very much, Ulrike. I want to start by saying that I really like using the OIG as like an informal venue to sort of get feedback on ideas and talk and really like have a community and discussion rather than sort of just presenting published work. This is going to be less of a report of completed research and more of a bit of perspective on where I think sort of my field of microscopy is going and some updates on what I, along with some teams here at Janilia and a lot of great sort of engineers and people from other labs and all that, what we're working on. All right, my personal focus in microscopy is on recording fast patterns of neural activity in vivo in opaque scattering brain tissue, so 2-photon imaging. And to do that, my lab has used principles from computational microscopy, and we've built microscopes that can adapt to the structure of your sample in order to improve imaging speed, mostly in our case, and signal to noise at that speed. So adapting to a sample implies that some component of the microscope is programmable in some way. And so I'm going to talk today a little bit about some of the hardware that enables that, how we've used it, and what we might be able to do with it in the near future. But first, I'll talk about some of these concepts like computational microscopy more generally, and then I'll go into sort of more specific examples from 2-photon imaging to quantify sort of what the benefits are and show you some of our approaches. Computational microscopy is a very big field, and it's becoming more and more difficult to define. So conventionally, it refers to methods that rely on computation to convert raw measurements in some form into data that we care about. So sometimes the data that we care about is an image. Sometimes it's something more abstract, like neural activity, for example. I guess the classic example of this is CT imaging, where the measurements correspond to different angular views of a different 1D, basically angular projections of a 2D sample. Another example is quantitative phase imaging, where we care about something that we can't measure directly on a camera, which is phase delay in an object, and so we need to use intensity measurements from a camera in order to recover phase. Another example is maybe multi-view deconvolution, where you take 3D pictures now of an object, but from different angles, and you want the best possible estimate of the underlying 3D sample. So this is a simplified diagram of that CT imaging problem. So, in CT, you have some lateral coordinate on your detector, and you rotate either your sample or your imaging system in order to look at that sample from a bunch of different angles. And then there's some reconstruction process, which is computational, to go from these 1D projections to a 2D slice. Another big aspect of computational microscopy is the design and optimization of optical hardware while taking into account how data is going to be analyzed. Whereas like conventional objective functions for imaging might be something like a spot size, like the resolution of an optical image, or an OTF. More recently, we've been seeing more model-based design that optimizes other objectives, like information about the location of something, or depth invariance, or what an image looks like after you deconvolve it computationally. A classic example of this is point spread function engineering. So there you can put an SLM or a phase mask into the detection path of a microscope to change its point spread function. And then you can optimize that point spread function for your different imaging tasks or sample types. Because a camera basically takes a 2D slice out of a 3D image that's made in the detection path of your microscope, moving information around axially in the Z direction is an especially sort of powerful thing that you can do by shaping point spread functions like this, and it's especially useful for 3D imaging. Srini Turaga's lab, sort of in collaboration with Misha and myself, we've been working on some of this for activity imaging, actually. But another important way that computation is used in imaging is the model-based control of optical systems, which is an idea that I think we can take advantage of much, much more. I think it's pretty untapped. I personally feel that if we can build more flexible optical systems and use computers to control them, then we can do much better at imaging tasks that we're currently doing, especially for activity imaging. For now, I just want to say that computational microscopy is a big field, and it's hard to sort of clearly delineate what is and isn't part of the field. There's all sorts of different ways that you can use computers and hardware together to do microscopy. I really like Laura Waller's definition of computational microscopy, which is that computational microscopy is the joint design of hardware and algorithms to solve problems that can't be solved by working on either of them alone. So there's really a synergy between the hardware and the ultimate processing that we do. I think in the last decade or so, we've seen sort of a convergence of the image analysis and hardware communities. So I think people on both sides have seen benefits of this approach, and more people are interested in doing this sort of merging. More microscopists are thinking about how to design instruments with the analysis in the loop, and vice versa. I think we've lured more and more sort of hardcore statisticians and modelers and machine learning folks into thinking about how to design microscopes. So one of the greatest strengths of computational imaging is that it allows us to use knowledge about a sample or an imaging system in order to make better measurements. Of course, computational imaging methods have been used to break a lot of long-standing limits in microscopy. I think the most famous example of this is localization microscopy. So, as you all know, optical images are limited by Abbe's diffraction limit. This was formalized in the 1800s. A lot of the optimization of lenses, was first done then to reach those physical limits. And I'm sure you know about localization microscopy methods like Palm and Storm. And these methods break the diffraction limit by using a prior. In localization microscopy, you take images of isolated diffraction limited spots, and then you identify the centers, with high precision, with much higher precision than the diffraction limit to tell you the location of the fluorophores, with the underlying assumption there that you're looking at isolated emitters. And even methods where the emitters aren't isolated, like SOFI, for example, actually still essentially rely on the same prior, and they need, maybe small numbers of emitters that are intermittently blinking. So there's actually an analogous limit on the temporal resolution of laser scanning microscopy. That's set by the fluorescence lifetime, which you can also break with priors. So fluorescence lifetime, sets this fundamental limit on how close you can make fluorescence measurements in time, which is analogous to the diffraction limit in space. Basically, when you excite a fluorophore, it takes some time for it to decay. And so let's say that you want to image a 2D plane with a 2-photon microscope, a sequential fluorescence laser scanning microscope. the number of measurements that you need to make point scanning is proportional to the number of pixels in the field of view. So that's n squared, where n is one side of your field of view, say. Of course, instead of illuminating one point at a time, you could illuminate multiple pixels at a time, so like a whole line at a time. And now you can, of course, scan that line across the sample and collect data from the entire sample plane with many fewer measurements. But now you have a problem because each measurement is a mixture of all the pixels in the line because of scattering. What we do to get around this is that in order to be able to computationally unmix those original pixels, we collect those projections at a few different angles. And so these measurements are actually just like CT imaging. They're different angular projections of the sample plane. So for example, to sort of continue the analogy to localization microscopy, if you have a bunch of point sources in your field of view, how does this look? So you scan each line across the sample, and each point source produces a bump of fluorescence at the corresponding location along that axis of the scan. When the line passes it, you get photons. So the locations of those bumps actually correspond to the locations of your point emitters on each of those those 4 different axes. Without a prior, it's actually impossible to recover the original image from such a small number of measurements. We have, a few thousand measurements and over 1 million unknowns. But if we have a good enough prior, we convert the measurements to obtain the original image correctly, and in the process break that lifetime speed limit. You'll have to trust me that we can reliably recover very high resolution images if we have good priors. So in this case, the image on the left required 1.6 million measurements, while the image on the right is essentially the same and it required just 4,000. So this type of imaging is called projection microscopy, and the microscope that we built that does this is called scanned line angular projection microscopy, or SLAP. So this is a movie, which you're probably seeing very intermittently, of particles flowing through a 3D imaging volume that was collected with SLAP. The volume consists of 100 planes, and there are 10 volumes recorded per second. Every plane contains about 1.5 million pixels and takes less than 1 millisecond to collect. So the recording is about 1.5 billion voxels per second. And that's 15 times faster than the fluorescence lifetime limit on raster scanning. So, just like localization microscopy breaks some fundamental limit on spatial resolution, this is now an approach that breaks a limit on temporal resolution.
Hey, Casper.
What's up?
Could I ask why the number of line is 4, not more or fewer? I'm Qinglun, used to work at Janelia, by the way, now in Taiwan.
Yeah, hi Qinglun. So there are trade-offs in the number of lines, basically. To collect more lines takes more time and is optically much more complex, but it makes the assignment, the unmixing problem easier. At the limit, so this is actually exactly analogous to CT imaging as well. So CT imaging, in order to do very good reconstruction without priors, you need a large number of angles. You need the number of measurements that you make to, roughly equal the number of unknowns that you're trying to recover. If you use fewer angles, so imagine the case of using 2 angles to recover the location of a bunch of particles, right? Like 2 perpendicular angles. Can you see me?
Yes.
Yeah. So if you place 2, let's say that you collected this angle, and you had 2 particles, right? You would get 2 bumps on each axis, and you wouldn't know whether you got this pattern or this pattern, right? And so actually 2 projections is not enough to localize multiple particles. You could only localize 1 particle that way. As you add more and more projection axes for this localization problem, the collisions between the projections, so the possible sort of false places where you might want to assign fluorescence and your sensitivity to noise in making those assignments decreases with an increasing number of projection angles. But of course, it takes longer to make the measurements, and it's much harder to build a microscope that produces such a large number of projection angles. So we actually, in order to be able to switch between illumination angles very quickly, we actually build out a separate optical path that produces each illumination angle. There's some tricks that we do to combine them, like and share scanning galvos so that we don't have to have a scanner for every path, for example. I think that gives you a sense of some of the trade-offs. For activity imaging, there's actually an optimal number of projection angles where doing more actually starts to hurt you because you pay more in shot noise. And that optimal number is around 8. And so we use 4. But it would be much more complicated to use 8.
All right, thanks.
Okay, so I mean, that was just sort of a cartoon sketch to sort of show you that models of your imaging process and of your sample can be pretty powerful for taking advantage for taking advantage of knowledge that you have about how your microscope works. And this is a core part of computational microscopy. A closely related but actually distinct concept is using data from your sample to tailor your measurements. And so for those two methods that I just showed you, localization microscopy and projection microscopy, nothing about those microscopes, nothing about the data coming in was used to change how those microscopes operate. But there are quite a few methods now that use sample information to do just that, to online control the microscope based on the data that's coming in. For example, you can use selective illumination in wide field microscopy or in one photon microscopy to only illuminate, say, some sources that you care about, and then not generate excess background or bleaching or damage to your sample. You can do adaptive optics, where you measure wave fronts coming out of your sample and use them to improve your point spread function, essentially. You can do random access scanning. This is very popular in two-photon microscopy, where again, you know where the targets that you care about are, and you basically only illuminate those targets by scanning between them and turning your laser off when you're not hitting them. Of course, there's sort of types of microscopy that fundamentally rely on getting feedback from your sample, like tracking microscopy. There's other forms of adaptive optics, besides the sort of pupil adaptive optics that are very powerful. Autopilot is a great example of that for light sheet microscopy. And there's also interesting super resolution methods and other things that rescue STED, for example, dramatically improves what you can actually do with STED imaging without bleaching your samples unnecessarily. All of these tend to be extremely effective techniques, actually, because they basically use information about your sample to not waste your effort, right? And so they tend to have minimal drawbacks. And there's a lot of overlap between these sort of adaptive and programmable microscopes and computational microscopy, but they're definitely not the same thing, right? So I guess what I'd say here is that the more flexible that your microscope is, the more you benefit by having a model of your sample. And conversely, the better your model of the sample, the more you can benefit from having a flexible microscope in order to optimize your measurements. So I think that these benefits are particularly strong in the case of two-photon activity imaging, which is the regime that I work in. And that's because two-photon activity imaging both has very strong constraints and you can take advantage of very strong structural priors. So two-photon imaging operates under strong physical constraints that push it in different directions. So on the one hand, you have heating, where the laser powers that we use in order to generate two-photon absorption are very close to the limits that tissue can tolerate. So the most that you can put into a mouse brain is a little over 250 milliwatts. And so these heating limits, they push us towards, in nonlinear microscopy, using tighter foci and lower repetition rates in order to get more nonlinear excitation per milliwatt of heat absorption. But there's other factors like nonlinear photo damage, bleaching, and saturation that are also highly limiting in two-photon. And these factors push two-photon imaging towards lower pulse energies and lower intensities. So these constraints are very strong. Actually, they're much stronger per collected photon in two-photon imaging than in one-photon imaging. I mean, you can consider any of these, but I mean, just looking at saturation, because we pulse illumination for such short periods of time, we have such short duty cycles over any given location in a sample volume, you reach saturation at much lower overall photon rates than if you can continuously illuminate your sample, for example, in wide field or light or some types of light sheet imaging. So these constraints, which are pushing against each other in different directions, they actually, they win out to different extents depending on your sample. So, photo damage and bleaching, they're most serious when you're imaging small areas, because you scan the same part of your tissue and the same fluorophores over and over again. But they're much less of an issue if you're scanning a much larger area, because that dose of damage or the dose of bleaching gets diluted over a much larger volume. Heating, on the other hand, doesn't care about your imaging area, and it's pretty much always a limiting factor because the length scale of heat diffusion in vivo is very long. It's about 3 millimeters in the cortex. And so imaging larger areas doesn't actually help you dissipate heat any better. It can be really hard to know offhand which constraints are dominant in which regime, but actually we understand these physical limits very well, and they can be easily modeled. Simulations are very good at understanding the trade-offs. And so I've learned, I personally have learned a fair bit by just performing very simple simulations of how multiphoton imaging works subject to all these different constraints. Signals are actually very sensitive to some imaging parameters, and that sort of reinforces to me the potential benefits of having computers and models control our microscope parameters. So as an example, you might think that because two-photon signal depends on the integrated square of light intensity, that two-photon imaging always benefits from using a tighter focus or a lower repetition rate. But in vivo, photo damage, bleaching, and saturation, they can all push back against that. And so for any given repetition rate, there's actually an optimal focus size that varies with imaging depth. So a focus smaller than the optimum, actually for that repetition rate, reduces the signal rate that you can achieve over the length of your recording. As you image deeper, the optimal focus, the optimal concentration of light in general gets smaller. So you want to have fewer, more intense pulses, or you want to have a tighter focus. At some point, of course, that optimum becomes smaller than the diffraction limit, and you become fixed at that limit. So when you reach this area, it's actually better to decrease the repetition rate, for example. And so knowing these things can be helpful for improving your signal level. So yeah, you can also keep the focus size fixed and look at what the optimal repetition rate in two-photon microscopy looks like as a function of here, for example, depth. And it varies by 2 orders of magnitude across sort of relatively standard imaging tasks. By far, the most useful parameter to control in two-photon imaging is where you shine light. So random access imaging dramatically improves achievable SNR. So these are simulations of high-speed two-photon voltage imaging. So random access imaging, of course, is this approach where you only illuminate a small part of the sample where you have productive indicator. And of course, in vivo or for activity imaging, we can know where those regions are in advance simply by taking a structural image. So this is what I mean by being able to take advantage of strong structural priors. I mean, there's other priors as well, or there's other ways that we can use models to guide structure-based acquisitions. Like if we care about certain types of correlations in measurements, we might want to make our measurements in one way. Or correlations in neural activity, we might want to make our measurements a different way than if we don't care about those correlations, for example. So random access imaging is actually a really large benefit for two-photon microscopy, and particularly in regimes where you have a small numbers of targets. And the main limitation of existing random access microscopes is the number of targets that they can address, as well as to some degree the types of patterns that they can make. Sometimes it benefits to make larger or extended foci of certain types. And the target rate issue with random access scanning comes from the access time. So, random access scanners are typically acousto-optic deflectors, and it takes time for a sound wave to cross the aperture of the deflector. And that access time, during which you can't really record anything, if you're going to a whole new area, is typically tens of microseconds. And as the number of targets increases, you actually end up spending most of your time hopping between targets and not recording them. The duty cycle becomes lower and lower. What's kind of cool is that in some applications, like voltage imaging of a few cells at a time, the access time just doesn't matter. As long as you can reach all of the targets, you can more or less compensate by increasing the repetition rate by basically delivering all your pulses during the short time that you are over the target. And so that's shown here, where if you optimize, the rep rate and the spot size really very close to the point where you no longer can even hit your targets, at least in principle, you can get close to the optimal signal rates. But for some applications, we want to access large numbers of targets with these conventional random access scanners. And so a project for me has been working to break the access time limit of random access microscopy. So the geometry of scan line microscopy gives us an efficient way to produce a random access scan. So we did this with our first projection microscope simply by putting an SLM in the imaging path, in an image plane as an amplitude modulator. So the SLM basically creates an amplitude mask in this case, and it blocks most of the excitation light from making it to the sample. It only lets light through in the regions that we actually care about measuring. So this in essence implements random access. It reduces background excitation. It improves our signal to noise or power efficiency. It reduces the degree to which signals have to be computationally unmixed as well. But the way that this was implemented in the first SLAP microscope wasn't very flexible, and it was limited by the fact that we're using an SLM here. Liquid crystal SLMs are really good for modulating monochromatic collimated light, but they don't work as well when you hit them with a range of input angles, like when you're using them in the image plane, and they also don't work when you hit them with a range of wavelengths, because each wavelength gets modulated slightly differently. So I know that Yoav came and talked to us a few months ago about using SLMs with light at multiple wavelengths. But I don't think that there are good solutions to this problem in the general case without very narrow band filters or else very small fields of view. Correcting this chromatic problem in SLMs for holography is a big area of research. And there are some ways to mitigate these effects in hardware, at least in principle, but those tools aren't available to us yet. And those strategies are actually, for the most part, only good for holography and not for amplitude modulation. But the most important point is that even the fastest SLMs can only update at a few hundred hertz. So if we want to image at kilohertz frame rates, it means that the SLM pattern is essentially static. We can't change the pattern on the SLM during imaging. DMDs, in contrast, have in some ways like an opposite set of advantages and disadvantages. Because they use mirrors, they do amplitude modulation that doesn't depend on wavelength, and it's very insensitive to incidence angle. And whereas SLMs work really well with coherent monochromatic laser light, DMDs, they have some special challenges with spatially coherent light. And that's because the little tilted mirrors on the DMD act as a Blaze diffraction gradient, meaning that only certain wavelengths of light can be efficiently modulated into a single diffraction order. And moreover, the light that's modulated by the DMD, it forms a tilted image relative to the new propagation axis. And it's difficult to image that tilted image into your sample at high resolution. So if you care about faithfully and at high resolution relaying an image from your modulator to the sample, SLMs are much easier to use, which is why we used one of them in our first SLAP projection microscope. But I always really wanted to use a DMD here instead, and it's mostly because of two benefits. And the first is the very high extinction rate, which makes it possible to select out very small regions of an illumination pattern without worrying about background light leaking through. And the other is the very fast update rates, which could give us a huge amount of flexibility in how we pattern light. And as I've tried to argue sort of earlier, flexibility is very important, or could potentially be very important for imaging performance. So, you know, whereas with an SLM, the SLM pattern has to remain essentially static. And so we can't change it as we scan in 3D. We can't change it in order to select out regions of the image. With a DMD, we can change the pattern on the DMD every time we scan a line across the sample. You know, to cut to the chase, we now have a microscope design that uses DMDs to modulate scan line illumination. And to use DMDs in this way, the biggest challenge was to solve these diffraction problems. So the heart of this microscope is this repeating module that we call a surfboard. It compensates for these diffraction effects and allows you to use a DMD in the path of a 2-photon microscope without suffering these effects. We compensate for them essentially by bouncing light off each DMD twice in a mirrored configuration. So the light comes in down here. It hits sort of a modulated field of view at the bottom of the DMD. It now passes through a one-to-one relay with the mirror here in order to be modulated again at the upper region of the DMD. And then it sort of comes back exactly anti-parallel to the input beam and is bounced up to be sort of further processed by the microscope. So this compensates for, you know, the image tilt that we would get if we just modulated light by this DMD and then used it out here. But it still allows us to modulate the light. So like that first slab microscope, we use a line of excitation, which in this case is a vertical line that scans horizontally across the field of view. But instead of having one SLM that modulates all the different paths, we now have a different DMD and just one scan line per DMD. And so because we can update these DMD patterns very fast and we have different lines and different DMDs, we really have a lot of flexibility in how we do imaging and how we pattern light, and we sort of have a lot of choices now for how we do computational imaging. So to start, we can create a raster scan by opening each line of the DMD one at a time, right? So as if we have a vertical excitation line that scans horizontally across the DMD for each of these patterns of open pixels, we essentially end up with a point that scans horizontally across that row of the DMD. The rest gets thrown away. So the result here, you know, is a raster scan of the entire field of view. We can also scan just sub-regions of the scan field, and then we get a local speed up in frame rate that depends on how many pixels we have in any given column. You know, under these conditions, some pixels are getting imaged faster than others. So this region is getting imaged at a certain frame rate. This region is getting imaged at a lower frame rate because there are more rows that it has to cycle through. And you can see that you can get these sort of inhomogeneous phenomena as well. But this microscope essentially has this sort of fixed, nearly constant bandwidth that can be flexibly distributed across the field of view to wherever you want. You can you can prioritize some small regions over others and image them at a very high frame rate while you sort of slowly cycle through other regions, for example. The real speed up of this approach comes if you can define regions of interest for integration. So, you know, integration ROEs, these are sets of pixels that we expect to fluctuate as a unit, like a single neuron cell body, for example, in activity imaging. When we analyze imaging data, it's very common to basically process one of these movies and integrate all the photons within these regions anyway. So doing that integration at the time of measurement can save us a lot of time. Of course, this creates a new challenge for us, and it's something that we're still working on solving, where we have to be able to detect and define these very large number of ROIs at imaging time, and potentially track them as well over time if we want to be able to re-identify things. We've been working with Jan Funke to develop these very sort of easy to train neural networks that can do instance segmentation like this, and that different people can apply to their different types of data sets. But there's other aspects of the problem that we haven't really worked on much yet. And so if anybody is interested in this, I would love to talk more about it. So yeah, I mean, different users have different types of objects that they want to track. My lab is most interested in synapses, and that's a little harder than segmenting cell bodies. But it's something that we've been working on. Okay, so what does it look like when you have these integration rows? I don't know how much you can see of this sort of fast blinking video, but if I pause it, you can see what's happening here, which is that this line, as it scans across this field of view, in any given column, there is only one ROI that's open, right? So this is a big difference from the original SLAP microscope, and it's made possible by having a DMD here. This means that the measurement at any given point in time only belongs to a single integration row, so you don't need to do any computational unmixing in order to recover the brightness of that region of interest. So we can essentially, we can actually quite easily produce images from measurements at video rate here. It also means that sparsity, which was very important for the original SLAB microscope, is not important for this type of microscope. It just, if you have denser objects, you open them up sequentially. There's no requirement for sparse labeling. So in this mode, what's actually happening is we're integrating each measurement along the line axis. We still get high resolution along the scanned axis, but we don't get high resolution along this axis unless you count sort of the edges of the regions of interest. And those sharp edges can still be used for alignment, for example, for motion compensation, but you don't get the details within the ROI along the vertical axis in this case. So it's trading resolution for speed just within regions where resolution doesn't really matter to us, which is within these regions that we want to integrate. And so there's algorithms that we've written that solve the planning problem for this, where you can specify constraints about how often you want to image certain ROIs, for example, and it produces an efficient sequence of DMD patterns that make this possible. So for the ROIs that I showed you in the previous slide, there were 500 regions of interest, and this microscope acquires those ROIs at a mean rate of 3.6 kilohertz each. And of course, we can reconstruct images or traces for those ROIs as fast as we record them. So the big limitation of this whole strategy is that it requires making this gigantic long line and then throwing away basically the majority of it at any given point in time. So you need to use a very, very powerful laser, and even still, there's a limit to how large of a field of view you can image. So, this was true to a large degree of the original SLAB microscope, and this is true of this new microscope as well. The field of view of this microscope is 230 by 310 microns, which is not great for doing sort of circuit scale neuronal cell body imaging, but I think it has a lot of applications in synaptic imaging or Drosophila imaging, for example, and single neurons as well. So one way to get around this limit on the field of view is actually to have these multiple DMDs that we can now steer to different regions of the sample. So in this case, we're using a remote focus system to we can decide basically where we put these different ROIs, and then we can steer them up and down using remote focusing actually quite fast, on the order of hundreds of hertz. Or alternatively, we can direct these two ROIs or these two fields of view to the same spot and do two-angle tomography. So this is reminiscent of the original SLAB microscope, where we're imaging essentially the same pixels now at two different angles, and we can do computational reconstruction. And in this mode, of course, you can do tomography at up to 20 kilohertz with this microscope. All of these different modes make this style of DMD-based imaging extremely flexible. So you can do, raster scanning faster than resonant galvos. I didn't really talk about it, but this microscope uses a polygonal scanner, which is very nicely matched to the sort of fixed field of view DMD style imaging. Polygonal scanners have a lot of problems for point scanning, but all those problems sort of magically go away when you do line scanning. So I think it's a really nice approach. If anyone has questions, I'm happy to talk about that. And that allows us to do extremely fast scans, faster than resonant galvos. And that allows us, and so, with this approach, we can do raster scanning at 20 kilohertz line rates. Or we can, we can use integration ROEs to get this sort of number of ROE dependent speed up in what we can image. But this is way, 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 way faster than sort of conventional approaches. It's both faster than sort of full speed tomographic slat microscopy, where we still had to do computational reconstruction. And in this mode, we don't have to do any computational reconstruction. And it's sort of off the charts compared to sort of conventional resonant scanners. And for large numbers of ROEs, as you can see, AOD-based microscopes simply don't work. So I can also imagine intermediate modes between sort of doing this sort of 20 kilohertz two-angle tomography with SLAP2 with this new microscope and this integration ROE case, so you could sort of fill in the space between these blue lines. And of course, it's because you have this ability to very rapidly pattern light, and in optogenetics, there's no constraint that things have to be unmixable. We can actually do optogenetics with a great degree of patterning specificity at extremely high rates. I mean, we can draw a whole new pattern to a DMD at 20 kilohertz and stimulate whatever sources that we care about there, updating it every 50 microseconds. So this is the sort of like strategy for flexible programmable microscopy that I'm pursuing right now, and I'm really happy to chat with people about what we're going to do with this and how it works. Thanks very much for your time.
So thank you, Casper. Are there questions out there? I guess we are still about the principle, how you maybe want to integrate this in your own system. Please feel free to ask Casper any types of questions about the different components.
I have a question.
Yep.
Hey, boys.
Hey, that's amazing. It looks really cool, and I really like the approach of combining the microscopy design and the algorithm. It's really obvious the way forward. One thing that, at least for calcium cell body imaging, that I would really like to see someone like you, super smart, to be working on, is to think of the next step of the analysis. So usually when we get calcium traces, people are interested not in actual calcium dynamics, but the activity that underlies it. And actually closing the full circuit, let's say that having a model of your brain, right, with the way that you labeled, and having, for example, when you select your ROIs, to me, it seems like you would also want to select some ROIs that don't contain cells to get an idea of the contamination that you get from neuropil signals, for example, right? And so how would you go about choosing those things, right? Would you choose one neuropil signal for each cell for each integration ROI that you do care about, or do you need less, or do you need more? I think those are really unanswered questions. And, if you can add another step to the modeling that would close that entire loop and tell you, given the firing rate, given the neuropil, given whatever it is that you care about imaging, right? How do you go about imaging? That would be the ultimate microscope that I would want to use, right?
Yeah, and what's your goal as well, right? I mean, the classic case here is, so you're talking about some more technical things that you would want to basically model and be able to control. But, let's say you're doing receptive field mapping, or you're mapping connections between neurons, right? There's a quantity that you want to know, and you have a model of what those receptive fields look like. I mean, already this is a closed loop process. Like the very first, sharp electrode recordings were done in closed loop. You get a cell, you like very quickly sweep some big patterns across the field of view. You figure out where it is. Now you start changing the patterns, right? Because it's impossible for us, we only have a limited amount of time with these samples, right? And it's not like Drosophila, right? We can't find the same neuron sample after sample and accumulate information in the same way. I mean, we can find the same circuit, we can find the same cell type, and we're making progress on that, but we're not going to find the same neuron doing the same computation, right? And so you have a limited time, and you have a huge space of possible receptive fields or connection patterns. I mean, these are combinatorial things that are just gigantic, right? And so there's a really important need for closed loop experimental design, where we know what we want to measure, we model our uncertainty, and we decide what stimuli we want to present, but maybe also what neurons we want to record from with our limited bandwidth at any given time in order to constrain our ultimate question, right? I think computers should be doing these experiments for us.
Yeah, I can totally see a multi-scale, you can zoom out and do low repetition to find interesting places, and then go in and validate your findings with very high spatial resolution imaging. If you can do that on the same experiment, that would be also pretty amazing.
Yeah, but that's very hard for a person to do at the time of the experiment. Yeah. Cool.
So are there more questions? Otherwise, so what's currently exactly limiting your field of view? You said that you are limited by 310 times 230 microns. So
yeah, I mean that's approximate, but okay. Basically, am I still sharing my screen?
No, you just stopped sharing your screen.
Okay, good. So we have to make this line, and the line has to, at every individual point after it makes it through the entire optical system, be able to perform two-photon excitation. We have to be able to have some, headroom over how much we excite, right? We want to be able to put a lot of power into a diffraction-limited spot. If we're going to decimate that line by a factor of 1,000, right? If we're going to put all of our power in a laser pulse to make a line, and then we're going to look at one pixel of it, speed to first order 1,000 times as much power in the laser. So this microscope uses a 56 watt laser currently.
Oh my God, I didn't know. That's quite, yeah.
Yeah, and it has a lower repetition rate, which makes things more efficient. And there's other things that help your efficiency with line scanning versus say isolated points, right? I mean, if you integrate having something that stays high for a long time and doesn't have a lot of edge, like a line versus a spot, helps with the integrated squared intensity. But to first order, you need much more power, right? So we use very, very powerful lasers, and it simply limits the degree to which we can decimate that line, right? So we can't make very long lines because we need 1,000 watts of power.
No, I understand. I was just not aware what the maximum, what your current power is of the laser, but that's, I think I've never worked with such laser before.
Yeah.
Anyway,
120
are there more questions?
56 watts. 1 megahertz if you want it to. So, I mentioned that we want to be adaptive in our repetition rate. That's super important. And so actually one of the so these are fiber lasers that we use. And I mean, if you've heard me talk before, I probably ranted during that talk about the possibilities of using inexpensive and powerful fiber lasers for microscopy. And we've been making indicators that work really well with these lasers and all of that. These lasers also have tunable repetition rates, which turns out to be a very important variable that you want to control for 2,4-10 microscopy, particularly as you change your imaging area or your depth within the sample.
Okay, so if there are no more questions, let's thank the speaker again. I think that was a great kickoff talk. So the next talk of our series will take place at the beginning of October. You will all get an additional email about who is exactly is going to speak, but it will very likely be a talk about image analysis. So stay tuned. I think, yeah. I'm looking forward to more interesting discussions with all of you. And if some of you actually would like to present during our series within the upcoming months, feel free to reach out to either Chen or me, or to Dimitri, Robert, and Christian at the EMBL. And yeah, so see you next time, I would say. Bye-bye.